Over 100 attendees from across the country came to the Ontario Marriott Hotel to discover the latest developments in liquid ventilation and other subjects related to cerebral resuscitation. The following seminar covers the advances in resuscitation from cardiac arrest and non-invasive induction of profound hypothermia, as presented by 21st Century Medicine's Director of Research, Mike Darwin.
My name is Mike Darwin. I'm Director of Research at 21st Century Medicine, Critical Care Division. And what I'm here to tell you about today is cerebral resuscitation, and that's going to segue into an enabling technology for cerebral resuscitation that we sort of had to develop if this thing is going to work and be practical. I've had a long-standing interest in achieving resuscitation. One of my very long-standing goals has been to achieve successful resuscitation after normothermic cardiac arrest. That means no blood circulation of the whole body, not just the brain, but the whole body, for 30 minutes. The current medical limit is about 4 to 6 minutes. I think we're well on our way to achieving 20 minutes, and that's 2/3 of the way there. And hopefully in the next couple of years, we'll make it to the 30-minute mark. With that said, I'd like to start out with some videotape of an animal that was subjected to 17 minutes of hypoxia and ischemia, 16.5 minutes of total cardiac arrest with no blood flow, and 30 seconds with additional seconds with no oxygen. And his name is Bob, and he is by no means the only animal that has been subjected to this procedure and recovered well. We have many others, some of which have been placed and are no longer in our hands, but Bob happened to be around and be the most photogenic, and so I will open with tape from that, and you can roll tape. This is Bob. Bob is one of our success stories here at 21st Century Medicine. Bob was legally dead, or clinically dead, if you want to use the medical term, for just under 16 minutes. And as you can see, he's in great shape. One of the things that makes dogs so useful for this research is that unlike other animals, dogs interact with people socially. They pay attention to us sometimes, and they exhibit curiosity, and they can read our facial expressions. This makes them very useful because if dogs are neuroinjured, they lose those special abilities. We exercise every bit of effort we can, and we're very successful at controlling both pain and anxiety, and our animals are both healthy and happy. Okay, could I have the first slide, please? One of the really critical things that we've learned that other researchers have not done in trying to recover dogs from cerebral ischemia. Ischemia, incidentally, is lack of blood flow at either normal body temperature or reduced body temperature. In this case, at 37.5 degrees Celsius. That's about half a degree above your body temperature. Dogs' normal body temperature is about 38. So other investigators in this field over the past 15, 20 years have compromised at 37.5. The investigative team that has done this work really has been just fantastic, and they deserve an enormous amount of credit, equal credit, particularly the co-investigators, because without those three people, the scientific insights that we've gained and the advances that we've made would not have occurred. Well, what are we talking about here? What are we actually trying to do? Well, we're trying to reverse and treat sudden cardiac death. That's a rhythmic death. It means an irregular heartbeat that interrupts circulation. It results in abrupt loss of consciousness and circulation, and it's not preceded by impairment of circulation. So we're not talking about people who are in the hospital with shock or dying slowly from cancer or something else. We're talking about people who have sudden cardiac death. And that results in global ischemia. And what that is it's absence of circulation to the entire body, failure of oxygen delivery, failure of substrate delivery, which is a fancy name for failure of delivery of nutrients that the tissues need, and failure of waste removal. The incidence of sudden arrhythmic death is surprising. It constitutes 540,400 cases per year, and it's 54.8% of all cardiovascular deaths in the United States, and that, incidentally, is the leading cause of death in the United States. Approximately 30% of the people who experience sudden cardiac death are 50 years of age or younger. And that's significant because these people are at the peak of their productive period in terms of their social value, if you want to value people that way. They have a lot left to contribute. And the principal cause of failed resuscitation in these individuals is cerebral ischemia reperfusion injury. It's not the inability to restart their heart. Well, what do we do for cardiac arrest today? Well, we have this thing called advanced cardiac life support, which is promulgated by the American Heart Association internationally. It is the American Heart Association, but the standards are applied worldwide. And they consist of CPR, early defibrillation, definitive drug therapy, and sophisticated post-resuscitation intensive care. The order has since been reversed on that, so that CPR now comes second and early defibrillation comes first. They tell you, call the paramedics first, don't start CPR first, call the paramedics, and there's a reason for that. It turns out that if you do CPR on somebody, the delay time and the time to ACLS have a great deal of impact on survival. And at 0 to 4 minutes, in terms of time to CPR, you get about 43% of the people back with a total of less than 8 minutes of arrest time. About 19% at 18 to 16 minutes, and about 10% at greater than 16 minutes. As you get out to 12 minutes, the survivals are negligible. However, I will have more to say on that in a minute as to what kind of survival you get. Survival is defined as discharge from the hospital. Current ACLS protocol, what do we do for people who have cardiac arrest? Well, we give them cardiovascular drugs, and there's a whole host of those that can be used, and most of those are aimed at restoring rhythm and maintaining blood pressure. Occasionally we will need to control their pH by giving them sodium bicarbonate if they've been in cardiac arrest for a long period of time. That's not a first-line drug anymore because it causes a lot of problems. But what's the real problem that people have in cardiac arrest? The real problem is that their brains are injured after 4 to 6 minutes of no blood flow. In fact, after 6 minutes, virtually everyone sustains serious neurological deficit. What kind of drugs are being used right now to treat people with that kind of injury? The answer is none. There are no ischemia reperfusion injury drugs in the ACLS protocol. In fact, there are none anywhere. So the current outcome of 4 to 6 minutes of normothermic cardiac arrest is about 20% survival with neurological deficit, and without neurological deficit, less than 1%. So that means that of the 20% of people who survive overall, and this includes people who arrest in the hospital as well, where the conditions are very optimum for resuscitation, fewer than 1% survive without significant cognitive impairment. Quality of life is very low. We're interested in this for a variety of reasons. We're interested in part because I was one of the founders of the company and one of the key early shareholders, and that's what I wanted to do. It's a difficult market to enter. It's not an easy market from a regulatory standpoint, but if you can enter it, it holds the potential for enormous profit. We're sort of the poor boys in 21st century medicine, even though we were the first ones to start out, because the cryobiology division has a lot more opportunity for near-term profit. But the opportunity to save and improve the quality of lives is enormous. We estimate that about 300,000 lives could be saved annually using this technology for the U.S. market alone, and that the projected revenues range from anywhere from $30 billion to $60 million annually on the basis of licensure alone. And the total gross revenues generated by this technology would be in the $3 to $6 billion range. We would, of course, not profit from all of that. Well, what were our objectives in doing this research? Well, they were to allow for routine recovery of patients who experience 15 to 30 minutes of normothermic ischemia. We call this ultra-resuscitation. To develop the drugs, hardware, and software to enable implementation of this technique. We want to facilitate widespread clinical application of ultra-resuscitation. And we want to make money, which is a perfectly reasonable thing to do as a result of this. Well, what causes injury and ischemia? I'm not going to spend a great deal of time on this. Saul has asked me to spend more time on the liquid ventilation cooling work, which has been a spin-off of this research. but there's a lot of things. Shifts in ion balance, acids forming, principally lactic acid as a result of no oxygen being present in the cells, injury to the mitochondria, the little powerhouses that make the cells, hyperglycemia, a lot of things cause injury. And the injury starts really quickly. In fact, the ionic shifts that begin to start injury occur in a 60-second period. What you see here, the timeline is missing from this graph, but this is 60 seconds after cardiac arrest. So the very most bottom trace is your EEG, and you can see that within the first 5 to 10 seconds, your EEG is isoelectric. You lose consciousness. And calcium, which is a big player in causing injury in cells, it turns on a lot of molecules inside cells that tear them apart. Begins to enter the cells big time and really almost goes to completion in terms of its entry within the first 10 seconds after the ischemic interval begins. When your body is functioning normally, it has oxygen available and glucose available. Glucose is a substrate. And you get about 36 ATPs out of this, and the ATP is used to regulate the cell volume, to run all the processes. It's the gasoline for the inside of your cells. If you don't have oxygen available, you run off of anaerobic metabolism and you get a lousy 2 ATPs as a result. And you also make a lot of stuff called lactic acid, which pulls your pH down and facilitates the entry of calcium into cells, and it facilitates the release of neurotransmitters that subsequently will cause a lot of problem, and I'll talk about that in just a minute. Two things happen in terms of shifts from the extracellular to the intracellular space. Outside your cells, you've got lots and lots of sodium and lots of calcium compared to inside your cells. In fact, you have 10,000 times the amount of calcium outside your cells that you have inside. And this disparity, if you will, is maintained by active pumping. It takes energy, it takes ATP to run little pumps in the membrane that are constantly pumping sodium and calcium out and pumping potassium in. And this is how cells regulate their metabolic processes and their volumes. So when you have ischemia occur, what happens is that you get a release of potassium, you get sodium and calcium coming in, which activate a whole bunch of undesirable processes, and you also get the release of two things. Most of you have probably been told, if you're interested in nutrition or health, that the body cannot store vitamin C. Actually, it does store vitamin C, and your brain is the principal repository for it. In fact, 80% of the vitamin C in your body is in your brain. And when vitamin C, which is called ascorbic acid or ascorbate here, is released by the brain cells in ischemia, it turns into a very nasty chemical. Actually, it is a very nasty chemical under certain circumstances, and that is when iron is around, and it turns out a lot of iron is released as well, you get something called the Fenton reaction going, and this generates enormous amounts of free radicals, which cause tremendous damage to the cells. You also get the release of glutamate and a bunch of other signaling chemicals that the brain uses. Nerves communicate with each other by releasing chemicals, and one of those chemicals is glutamate, another is aspartate, another is nitric oxide. There are a whole family of them, and these are released in enormous quantities by brains that are ischemic because, again, it takes active energy to maintain these things in little vesicles and the little balls that you see there that they're normally stored in. Well, what are the consequences of all of this? Well, the ion shifts, as I've alluded to previously, cause a very serious disruption in terms of the metabolic effects downstream, but they have a more immediate effect. The potassium moves out and the sodium moves in, and what we have here is a normal situation with red blood cells sitting in a capillary. That's what you see. Figure out how to operate this thing. That's what you see right here. This is a capillary. Those are red blood cells in it. And in ischemia, because you do not have any energy to pump water or sodium out, the sodium comes in and water comes with it, and the blood vessel ends up very dehydrated with the red cells really packed in there very tightly, and the colloid, which is like a gelatin that is present in your blood, becomes very thick and viscous, and we call this hyperviscosity. And what you see here is an electron micrograph taken from an animal that has been extensively washed with a clear solution after a very brief period of ischemia, 6 minutes in fact, and you see typical packing of red cells that have plugged up this capillary, and all the tissue supplied by this, all the cells supplied by this are not going to get any flow because of this big plug of viscous red cells and protein. albumin. And the red cells also become stiff because they need ATP to function as well, and when they run out of ATP, they get very stiff. This is significant because a capillary is only about one red cell wide at its smallest point, and the red cells actually have to squeeze and bend and deform to get through them. What you see here, and this micrograph took a lot of work, this was made by Jerry Leaf, a former colleague of mine who now unfortunately is no longer with us. This is the surface of the pia, which is the blood vessels that cover the brain in a normal animal with perfusion going on. And what you see is just red cells moving through there at breakneck speed. These are capillaries, and you can see some that are out of focus in the distance there. This is the same tissue, same spot, after restoration of blood flow following 6 minutes of ischemia. So with a normal mean arterial pressure of about 60 millimeters of mercury, this is what you get in the first few minutes after reperfusion. In fact, this was taken 15 minutes after reperfusion started. So that's what hyperviscosity is, and it's a major problem. There are a lot of biochemical problems, and I won't spend a lot of time on these because of the time constraints today. Free radicals are one, and there's a whole bunch of them. And one of our fortes at 21st Century Medicine has been realizing that other free radicals than this one, and this one, superoxide and the hydroxyl radical, that there are other radicals like nitric oxide and oxygen radicals that are, and peroxynitrite in particular, that are really important players in ischemic injury, as well as singlet oxygen and hydrogen peroxide, which have been understood for some time to be damaging. But this one, peroxynitrite and nitric oxide, these were not generally understood to be tremendously damaging to the brain. And we have a whole panoply of pharmaceuticals that we've developed to moderate injury from that. So we're taking these free radicals and we're taking these free radicals that are largely generated in the little powerhouses of the cell, the mitochondria, and we're going to be blocking those with drugs. I'm not going to spend much time on this other than to say that the time course for most of the acute injuries is within the first 10 minutes, and then you get things like hyperglycemia, which occur over a period of hours after you start circulation again. We call that reperfusion. You get excitotoxicity, which is the brain cells firing madly as a result of those neurotransmitting chemicals being released. And most importantly, perhaps, you get activation of the immune inflammatory cascade. And and as a consequence of that, you ultimately get apoptosis. And apoptosis is programmed cell death. I want to pause for just a second to explain the immune inflammatory cascade to you. If you are injured in the natural world, the normal response is to have inflammation occur. You have all cut yourself or sprained an ankle or burned yourself, and what happens? The area immediately gets red, swells up, hurts, and if you have any sort of bacterial entree into the wound, you get a bunch of white cells there and you have pus. And the reason for that is that we are constantly surrounded by things that want to get inside of us and use us, little microparasites that want to use us for food. So the normal defensive strategy is to just kill the whole area surrounding the injury. And that's a great strategy. It's worked very well. That's why we're here today. As long as the injury is local. If it gets too big, if it's a whole arm, then you die. And if the injury is to your whole body, then your white blood cells and hundreds of other cytokines and parts of your immune system that are normally there to defend you turn on the damaged tissue in your entire body, and you sort of self-destruct from the inside. And understanding the importance of the immune inflammatory cascade has been critical to our success. So I want to go back to that slide. So the purpose of this work was to develop a controlled model of sudden cardiac death, to find out what the real causes were. We call it pathophysiology of the damage, to develop a reproducible systemic and cerebral resuscitation model, and most importantly, to achieve consistent long-term survival without neurological deficit. There are tens of thousands of papers out there that show all kinds of good results in a single animal model with a single drug intervention where the animals don't survive and their neurological status is not evaluated. We're not interested in that in terms of the work we want to do. We're building on that work. We're taking that work and applying it. What we want to see is the whole animal survive. And that's critical because very few people are doing that kind of research, can afford to do it, or want to do it. Most academics focus on a small part of the problem. They want a nice publishable paper and they want nice clean data. We want clinical results. We want something that saves lives. So Why did we pick dogs? Well, I've already given you one reason. They're socially very responsive, and if you injure them neurologically, their social responsiveness goes away. Their response to ischemia reperfusion injury really closely maps our own. The timelines for damage, irreversible damage, and serious damage are almost identical to that in humans. They're obviously sex, weight, age, and breed are more controllable. Captured primates, actually, ones that swing from trees, which are most of the ones available, have more resistance to ischemia than either dogs or people. And there's a reason for this, and I'll explain it. There's a large pre-existing database with this species. Dogs have been the model for this work for the last 20 years in terms of large animal survival model. And we can do in-depth neurobehavioral evaluation on them, including determine whether or not they can interact with us socially, which is really important. Pardon? Why are dogs much more resistant than monkeys that brachiate from trees? Well, this is a dog head in cross-section. And most of a dog's head is not his brain, which is right here. It's this football helmet of muscle that he wears. And if we look at it in cross-section the other way, you can see that a dog's brain is extremely well protected. And what this means is in the natural world, dogs rarely get head injuries. In fact, they virtually never get head injuries. And humans rarely get head injuries in their natural environment compared to animals that swing from trees. If you brachiate from branch to branch, you're going to fall and you're going to hit your head. So it turns out that macaque monkeys, for instance, have about a tolerance of anywhere from 15 to 17 minutes of ischemia, cerebral ischemia, with fairly good survival and neurological outcome without any intervention other than resuscitating them, whereas dogs are completely fried by 10 minutes. So this is a very good model for us. It's also a good model because we have large volumes of blood and urine, and we have large blood vessels to work with, which allows us to do cardiopulmonary bypass. And I'm not going to spend a great deal of time on this, but I will point out that the survival rates and neurological deficit times are very close to that of humans. By the time you're out to 15 to 30 minutes, you have 0% survival with uniform brain death. And at 30 to 60 minutes, you get what's called panorganic death, which means you have real difficulty resuscitating the animals. And you fail because almost every organ system has been injured by ischemia. This slide I won't spend a great deal of time on beyond pointing out that the current absolute limit on reversibility that the medical community, the leading edge of the medical community, considers possible is 60 minutes. And our objective is to aim right here for the so-called uniform reversible change zone, right in the middle of that, which is 30 minutes. Most of the current animals have been done in the 15 to 17 minute range. These are pilot studies. We hope to get back to doing this work and starting our statistical run probably in about March. We'll restart the work probably in January and February. The protocol basically consists of causing cardiac arrest under anesthesia at 37.5 degrees Celsius, allowing that arrest to persist for 15 to 17 minutes. Then we reperfuse them using a heart-lung machine. We dilute their blood by about 25 to 30%. We rapidly cool them to 32 to 34 degrees Celsius. This is an older slide. And we use multimodal drug treatment. We take over regulation of their metabolism and provide a lot of support. We control pH, we control their blood gases, we control their glucose, calcium, and potassium levels. We elevate their blood pressure as much as we can, usually to 140 to 190 millimeters of mercury mean arterial pressure. We minimize perfusion defects. We nearly inhibit the immune inflammatory cascade with a whole host of drugs. And we abolish post-ischemic systemic edema by inhibiting nitric oxide synthase, which is inducible nitric oxide synthase, which is a molecule that has only recently been understood to be a key player in ischemia injury. We also inhibit the excitotoxicity that comes from release of all those brain neurotransmitters, and we rapidly restore that cellular gasoline level, the ATP level, to a high level using a novel drug. This is what a typical experiment looks like. Animals on the table, computer stack and array for data acquisition, animals being mechanically ventilated and is under anesthesia. We have now computerized data acquisition and limited computer control of the whole process, including nearly automated control of bypass so that we don't have to have a perfusionist anymore. Why do we use bypass? Well, the answer is simple. If we were going to be using CPR followed by defibrillation, we'd have varying times before we could know that we restarted circulation cleanly. So if we want a nice clean end to the period of circulatory arrest, the best way to do that is with a pump and oxygenator, because we can turn it on, and we know the instant we turn it on, we're ready to go. It permits total control of blood flow rate, it allows uniform distribution of therapeutic drugs, and it allows rapid post-induction of mild hypothermia. If you cannot cool that critical 3 to 4 degrees Celsius in the first 15 minutes, the animals will not survive. You have to be able to cool that rapidly. And of course, it facilitates control over vascular volume and central venous pressure. This is the typical setup that's used. The heart-lung machine, as we know it, is gone, replaced by a computer console array and monitoring equipment. The basic circuit is very simple. We're running femoral bypass, which means we steal the blood from the femoral vein. We run that blood back through a compliant reservoir, through a pump, through an oxygenator, and then back up retrograde to the animal. This is a perfectly workable technique. This is the circuit set up and running with blood during reperfusion of a dog. Oxygenator, compliant reservoir, filter, and pump. Dog on the table. We achieve cardiac arrest by passing 2 spinal needles under the skin on either side of the heart and administering 110 volts of wall current for 1 second. This is an example of something that is a black art that took us a long time to figure out. The leader in this area of work prior to us is a fellow named Peter Safar, who is a very brilliant and fine scientist who is now largely retired, and he developed the model that we're using of arrest. And we had tried cardiac arrest using his published protocol again and again with alternating current and had terrible results. We couldn't get the animals to arrest. Finally, I went and spoke with Safar, and during the course of our conversation, I explained the problem, and he just smiled wanly, and he said, "Ah, yes, we never published that. You see, the key is you have to put spinal needles under the skin so that the current passes uniformly across the heart, and then all your problems go away, and you'll get arrest every time after 1 second." We've never had it fail. We've done 20 dogs since then. What are we doing? Well, we're using hemodiluent, which is a dextran 40 containing solution, to drop the viscosity of the blood so that we can get over that thickening problem. We're giving drugs to raise blood pressure, drugs to control pH, drugs to control glucose. We're using 6 drugs to control free radicals. We're using 3 drugs to inhibit excitotoxicity. We're using 1 pretty poor calcium antagonist drug. That's not a mainstay, actually, of our therapy. We're blocking the things that calcium activates downstream. One of the reasons we don't mess with calcium too much is that if you block it, most of those drugs drop your blood pressure and interfere with cardiac rhythm. So we've sort of stayed away from the calcium channel blocking approach, which has been the mainstay, incidentally, of most other researchers' approach to inhibiting ischemic injury. Bradykinins, leukotrienes, and COX-1 and COX-2 are all inflammatory things that are released and cause tremendous damage, as are phospholipases. We block all of these. We block platelet activating factor and platelets themselves, and we inhibit something called PARs, which I won't spend any time on, and we have 2 drugs to rapidly restore ATP levels. This is transcranial Doppler, which lets us look inside the animal's head and look at what the blood flows are. And this is a normal animal before anything is done to him under anesthesia. And you can see the brain has a lot of luxury perfusion between diastolic beats. In other words, between systolic beats. This is the heart contracting, blood is going out. But even between heartbeats, these 2 peaks, there's a lot of flow going on in the brain. The brain is almost alone in organs for having that much luxury flow. This is an animal after reperfusion and ischemia, and this is 1 hour after the ischemic episode. And you can see that the flows are astronomical. If you slap yourself on the arm, or you sprain your ankle, or you burn yourself, you probably notice that everything gets real red and hot, and that's because the blood flow increases. And this is a marker of how much is going on in the brain in terms of blood flow. Now this is significant because you see normally in a control animal, or in most situations, at 1 hour, the blood flow has almost gone away completely because the brain has experienced something called no reflow and has died, and there is no longer any perfusion or circulation going on. Our animals continue to exhibit elevated flows. This is, I believe, the 3-hour mark, and this is 4 hours. And finally, after about 10 hours to 12 hours, we go back to something approaching baseline, and then we are at baseline at about 15 to 24 hours post-reperfusion. And if the animal's going to survive, that's pretty much what his transcranial Dopplers look like. Our survivors, one of the interesting things is we have tried to get the mean arterial pressure to the level that Peter Safar and everybody else believes is absolutely critical to survival, and that's 140 to 150. And that's really interesting and an important point because you see, if you've had a heart attack, part of your heart has been damaged. And to get your heart to make your blood pressure go up to 140 or 150 and hold it for 2 hours is almost impossible. So that's been a major barrier to application of this technique. However, what we found is that a significant number of our animals who survive don't achieve these very high reperfusion flows. In fact, they achieve far lower flows, and yet they survive without neurological deficit. Our non-survivors have no significantly different flows in the experimental group than our survivors, and our controls actually do better in terms of their mean arterial pressures than our experimentals. And that's because some of the drugs we give drop blood pressure a bit. So this is very important because now we've got a technique that allows us to not have this tremendous need for high blood flows. Our short-term adverse effects, I won't spend a lot of time on this, but elevated liver enzymes, a huge secretion of neutrophils, which we're working on getting rid of. This problem is solved. Profound diuresis. Cortical blindness is largely solved. Cognitive impairment of 1 to 6 weeks is largely solved. Hyperglycemia is under control right now with insulin. And we have a profound decrease in vascular capacitance, which is a necessary side effect of the drugs we're using. The profound hypokalemia, which means lack of potassium that we see in the animals due to loss through pumping of potassium into the cells, remains a problem, but it can be treated by giving very large amounts of potassium. It is a side effect of one of the drugs that makes the cells have a lot of energy recovery quickly. We see transient VQ mismatch in the lungs, probably as a result of the vehicle that we're using to give these drugs on, which is a special micelle, a special fatty molecule, or special fatty package that the molecules are packaged in that shuttles them across the blood-brain barrier. And we see profound skeletal muscle weakness, cerebellar ataxia, and isolated PVCs, the cardiac arrhythmias with occasional runs of VTAC for about 1 to 3 days. All of these changes go away, usually by the 3-day to 1 week mark. I've only picked one enzyme. There was a lot of data in the previous presentations. I've cut out most of the scientific data here because of time. This just shows that our controls and our experimental non-survivors experience significant rises in enzyme levels. One important thing to keep in mind is these are peak levels achieved. The controls do not survive beyond 48 hours. So for a lot of their enzymes, which are markers of damage, there are things that are, these enzymes, well, LDH in particular, is something that is released from cells when they're damaged, when they're broken or busted. These animals probably never reach their peak enzyme levels because they don't live long enough, whereas our survivors do. One of our non-survivors up here, we did not we had tremendous elevation of LDH, and we're not sure why that's the case. This is not easy work to do. Each experiment costs between $10,000 and $15,000. The staffing that is required to care for the animals is enormous. This is Joan O'Farrell, who is our data acquisition and quality assurance person. Joan is invaluable. She conducted a lot of the trials for Prozac and Xanax for the pharmaceutical companies, Lilly and Upjohn. And she really keeps us in line there and has improved our data acquisition enormously. This is ICU. This goes on for anywhere from 3 days to 10 days, and it requires round-the-clock, 12-hour shifts of skilled people to care for the animal. This is almost immediately after the arrest period. This is probably about 6-7 hours after cardiac arrest. The animal is heavily sedated, not conscious, not aware of his surroundings, and on something called a T-tube. He is breathing on his own, but he's getting supplemental oxygen, and he cannot defend his own airway, so he has an endotracheal tube in. And this is probably about day 3. The animal's awake, beginning to respond to us and wake up. Well, how have we done so far? We have some exclusions. We had a learning curve on this, and we learned that you have to really control mean arterial pressure well. We had one animal that we had terrible hypotension on as a result of administering one of our experimental drugs too fast, so we excluded that animal. We had one animal whose lungs we blew as a result of elevating his central venous pressure to 60 millimeters of mercury. Normal is about 5. This again was a drug side effect that we learned about when we added a novel drug. We made a mistake which cost us one animal in terms of mixing up the prime that we use. We had an unstable drug preparation, again as a result of a change in protocol. So these animals were excluded from the data that we're about to discuss. In our controls, we have done a total of 4 basic controls. All of the animals died, 100% mortality. We did one drug control, which was no ischemia with the full drug protocol and cardiopulmonary bypass. The animal survived with very rapid recovery and no neurological deficits, and it was a long-term survivor. This is a bad slide, but basically this is our survivors, and what we have is very, very good outcome as a rule. Excellent neurological, acute neurological recovery in one animal, poor to moderate in the other animals. By acute, we mean the first 96 hours. I can now say that short-term recovery, which we consider 6 weeks, we had one excellent, a good, actually, excuse me, 3 excellents and 3 goods, one non-survivor and another non-survivor up here. So we've got about 75% survival overall. The adverse long-term sequelae, we see mild cognitive impairment in about 3 of the animals. It's nothing you would notice if you were to see them, but we notice it because we have pre-baseline on these animals and we know how they behave socially. Decreased social responsiveness to humans in 2 of the animals. So overall, we have about 50% of dogs experiencing mild, lasting cognitive defect, and the other 50% were normal, indistinguishable from control. One area that was, until very recently, in fact, it was the last couple experiments, an unresolved problem for us was the cerebellar ataxia. The animals behaved like they had cerebral palsy for about the first 2 weeks after the procedure and then recovered completely. And when we looked at their brains, we found hundreds of very small hemorrhages, which you can barely see as just dark areas here in the cerebellum, the part that controls the motor area. By contrast, the frontal cortex, which is sectioned right here, has no hemorrhages at all. The control animals are loaded with hemorrhages throughout their whole brain. We have since solved this problem by using a nitric oxide synthase inhibitor, which is transported into the brain on micelles, and it took us a long time to figure out how to do that. Our current limitations on this protocol are that we have moderate to severe liver toxicity. We have a profound decrease in vascular capacitance, requiring that we actually drain off about half the animal's blood volume for about the first 2 or 3 hours after we restart circulation. If we're not careful, we have hypotensive effects of giving these drugs, and that requires that we do automated control of rate and timing of the delivery. We're going to be immunosuppressing these animals without any question, although we have not seen any significant problem with infection. We did have one animal die of infection. And we do think that we have possible increased difficulty in achieving defibrillation, although we're not really sure about that. Our consistent run, our statistical run has not been done, and we don't have enough consistent animals to be certain of that. We definitely have profound and sustained potassium, low levels of potassium in the blood as a result of the drugs we're giving. And this is the best results with Peter Safar's model. He achieves 100% survival in both acute and control groups with about 11 minutes of cerebral ischemia. This is 11 minutes. 50% of the hypothermic group that he treats with rapidly induced hypothermia and cardiopulmonary bypass reach an OPC1 level, normal level. 60% reach OPC2, which is moderate disability, and moderate disability on our scoring system would be severe disability, meaning the animals cannot even walk. 42% of the controls, that's animals kept at normal body temperature, reach an OPC2 level, which is moderate disability, and 50% reach an OPC3 level, which is severe disability. Those would be animals that would be completely unresponsive and bedfast. And he sees pronounced multifocal neuron loss in both his control and experimental groups. He has done some dogs at 17 minutes, and none of those dogs survived long term. The best he was able to do was moderate disability, which on our scoring system would be severe disability, and One dog in each of those groups dropped back to what he calls severe disability, which would be really bad in our group. So he had one dog in each group reach that at 96 hours. That is not a long enough follow-up time in our estimation. We have some defects in this model. We're giving the animals heparin pre-insult because the heparin-bonded catheters are very expensive. We don't think this is a major limitation on the model based on Safar's work and others. We don't think clotting is a major problem during the first 15 to 17 minutes. We are using a barbiturate for our anesthetic. We taper that off near the end, but there's residual barbiturate, and some people have shown that to be protective, although not very protective, certainly nothing to give us the results we've gotten. We do not have systematic evaluation of pre-insult learned behaviors, and we do not have systematic evaluation of post-insult learned behaviors. We've actually done a lot better on this since the slide was prepared. We do not have quantitative histopathological evaluation of the brain or other organs to show us how many neurons have been lost, and there have been significant variations in drug protocols amongst the animals. We're not blinded, and we have a relatively small number of non-drug controls. However, there are actually a relatively large number of non-drug controls done in other laboratories, and that all dogs die after 15 to 17 minutes of ischemia. So what are our advantages? Well, we have 100% acute neurological recovery. That means animals wake up and respond and are conscious after 15 to 17 minutes of whole body normothermic ischemia. 70% long-term survival following 15 to 17 minutes of ischemia, 50% survival with absolutely no detectable neurological deficit, and 50% survival with very mild cognitive defects. In other words, they have normal kennel behavior, they look normal, they act normal, but their ability to learn and interact socially may be impaired mildly. Well, what are the technical requirements for this? I'm going to skip number one, but just say that you have to be able to cool very rapidly, and that has been the barrier to applying this kind of technology, the need to induce hypothermia rapidly. You need to be able to defibrillate in a very timed fashion. You need to deliver your antiarrhythmic drugs in just the right sequence and time. You need extracorporeal support currently, which is not something you can do in the field. And that's a lot. What you get if you have all these things is healthy, happy dogs. And I can tell you that being dead for 17 minutes does not stop you from reproducing, because this is Cerberus, one of our survivors, and this is a litter of about 8 pups that she had. And she, in fact, gave us 2 litters. So we know that dogs can reproduce, and another of our dogs named Claudia also gave us a nice litter of pups. This is Maud, one of our survivors. Maud has been placed and is currently living in Phoenix with a lady who operates a kennel there. And I just talked to her a few days ago, and she said Maud is doing very well and is very sharp and has learned the rules of the house. And while she doesn't get along well with other dogs, she does great with people. I'll go over this very quickly. We got a lot of barriers to making money with this technology. Western medical regulatory agencies, including our archenemy, the FDA, rarely, if ever, approve multi-drug cocktails. It's difficult to get regulatory approval for sophisticated biomedical software. Think about it. Running a heart-lung machine isn't that complicated, and yet there are about 20,000 perfusionists in this country, and they make their living running heart-lung machines. And computers could do it better, and yet there's no system out there for running heart-lung machines yet. You need a multimodal approach to delivering all of these drugs, and that multimodal approach is very counter to critical care medicine, and because it's going to be automated, it may make a lot of people edgy, and it raises a lot of liability questions in terms of software. You need to deliver multiple drugs within the first 5 to 15 seconds, and you need to sustain this delivery, and you need to get them across the blood-brain barrier, and you need a unique technology to do that, and you need hypothermia. You have to cool to 32 to 34 degrees Celsius within the first 3 to 5 minutes, and you need artificial intelligence to do all this. So that's our results to date with this model, and that is what we're out to do. 21st Century Medicine was founded approximately 5 years ago, starting with the Critical Care Medicine Division. It's the mission of the critical care division of the company to develop technologies to allow for prolonged resuscitation after cardiac arrest. Our aim is to be able to successfully resuscitate people following heart attack, drowning, for as long as 30 minutes after cardiac arrest has occurred. The other primary mission of the company is induced hypothermia. We're developing methods for non-invasively and rapidly cooling patients to 3 to 4 to 10 degrees Celsius below their normal body temperature using perfluorochemicals as a liquid ventilating medium. This allows us to use the lungs in a non-invasive fashion to move cold liquid in and out of the patient's body, and in so doing chill the circulatory system and thus the patient's entire body without having to use surgery or other kinds of invasive technology that would be time-consuming or impractical to apply in the field. We have made great progress in this area, and our facilities are at or better than that of university level. We have state-of-the-art blood gas equipment, computerized data acquisition, which allows for real-time management of data and fast interpretation of what has occurred. More important than equipment is the staff. The quality of people 21st century medicine has that has made this work possible is truly phenomenal. Without them, the kind of progress that has occurred would not have been possible. I've talked a lot about hypothermia and the need to induce it quickly, and this has been a problem that I have confronted in my career for 30 years. And it's one that, I'll need a second slide tray now for that. It's one that I've had great difficulty with. The first and most obvious way to induce hypothermia in somebody is to pack them in ice. And that's done clinically in medicine. It's not very effective. And the next most effective thing is to immerse them in a stirred ice water bath, and that's somewhat more effective, but it's not very practical. It takes about 300 pounds of ice and another 200 or 300 pounds of water. So that's not something you can do in the field very easily. And this has been a major problem. Now, there is a really good way to induce hypothermia very quickly, and that is you can hook somebody up to a heart-lung machine. And there are actually ways of using needles to access somebody's femoral artery and vein and hook them up to a heart-lung machine. And under ideal conditions, with somebody who is not in cardiac arrest, where they have guide wires in already, you can get somebody on a heart-lung machine on bypass in about 15 minutes. Well, why are we doing this? Well, we want to treat hyperpyrexia, which is elevated body temperature, occurs in heat stroke, infectious diseases, and drug reactions. And there are a lot of cases of this, about 50,000 a year in the United States. Turns out that the longer you're exposed to the high body temperature, the more injury that you have and the less chance you have of surviving, which I guess is no surprise. We want to prevent post-ischemic encephalopathy, which is a fancy way of saying we need to cool people really quick after they have a cardiac arrest, then we resuscitate them. We want to reduce post-traumatic encephalopathy following head injury. Right now there is a huge multi-center randomized clinical trial going on for closed head injury with hypothermia, and that is showing spectacular results. And their criteria for entry into this protocol are that you be cooled by 3 degrees Celsius within 3 hours of the time you have the head injury. Now, how many of you have burned yourself and put your finger on ice or the burn on ice and had tremendously good results with that and not had a blister form? A lot of you, right? But if you wait, say, 3 hours to put ice on, what kind of results are you going to get? Not very good. So clearly, the more rapidly you induce hypothermia, the better. And then sort of a wild card, long shot use of this technology is the induction of what Peter Safar calls short-term suspended animation. I call it the Princess Diana scenario. You have somebody who's bleeding out or has terrible traumatic injury, and you could save their lives if you just had more time, if they were just colder and slower in their metabolism, because cooling slows metabolic rate. But currently, we don't really have any way to cool people fast. External cooling, the best you're going to get is either 0.01 degrees to 0.2 degrees Celsius a minute, and it has a lot of problems. As soon as you start to cool somebody by packing them in ice, what happens? Their blood vessels constrict. We all know what it's like to go out in the cold or be in cold water. Your response is to constrict your blood vessels, and that slows the rate of cooling. It also cuts off blood flow to the tissues that are vasoconstricted, and that means that they become ischemic. Bypass is invasive, it's time-consuming, it requires anticoagulation, which if somebody's injured causes bleeding, and it's technically demanding, and it is above all not portable. And peritoneal pleural and pleural lavage again require skilled people, they take time, and they're very ineffective and slow, particularly in the presence of vasoconstricting drugs, which we have to give to achieve resuscitation. Well, what about other methods? About 4.5 years ago, I thought about using the lungs as a heat exchanger. And I thought, this is a brilliant idea. All of your cardiac output flows through the lungs. They have a huge surface area, about 70 square meters. You don't need any brilliant technology to implement it. If you can put a tube in somebody's windpipe, you can probably use the technique to do it. And we know that perfluorochemicals, they're basically organic molecules where all the hydrogens have been substituted with fluorine, perform very well in the lung. They don't damage the lung, and they can carry oxygen and remove carbon dioxide. So this seemed an ideal thing to me. And I went off to patent it. And I discovered, to my dismay, that somebody else had thought of this before me and had patented the approach of using total liquid ventilation. I was replacing all the air in the lungs with fluorocarbon. And the lungs have an enormous surface area, and this should work really well. Well, we had already had some experience doing total liquid ventilation, and we found some problems with it, which I'll talk about in a minute. But before I do that, I'd like to once again revisit the various methods that are available and talk about the method we've developed in terms of the time to application. There's external cooling, which is immersion in 0 degrees Celsius water, and that's going to take about 80 minutes, assuming that it takes you about 15 minutes to reach the hospital, if it's available. I've worked in 3 hospitals, 2 of them major medical centers, and I've never seen ice water baths available. People are cooled with ice packs and a cooling blanket under the best of circumstances. External cooling using a hypothermia blanket is going to take you about 130 minutes to get to a 5 degrees Celsius temperature drop. So that's not going to do you much good in heat stroke or in post-cardiac arrest encephalopathy, because you have to get the temperature down within the first 10 to 15 minutes if the patient's going to survive. Cardiopulmonary bypass. The perfusionist that we used to employ almost continuously for our dog work stopped in to see me when I was working on this slide, and he saw this 60-minute figure here, and he just laughed. He was laughing. He said, "My God," he said, "where'd you get that at?" I said, "Well, I'm trying to be generous." And he said, "Well, you know, the best I've ever seen anybody do emergently coming in from the ED is an hour and a half. And I think at the centers that are equipped for this, which is very few, probably no more than a dozen in the whole United States, 90 minutes is probably the absolute fastest anybody has gotten on bypass in practice. So these things are not very practical for quick application. With mixed mode liquid ventilation, we can be down 5 degrees Celsius in 30 minutes. In fact, we can be down 4 degrees Celsius in about the first 5 minutes. And all you need to do is intubate. Well, let's talk about liquid ventilation. The first kind of liquid ventilation that was envisioned and developed was total liquid ventilation, and this goes back to the 1950s when a guy named Kilstra, who was interested in deep-sea diving, started experimenting with hyperbaric saline in dogs' lungs, and he actually had dogs survive being ventilated with saline for a short period of time under hyperbaric, and there's very high-pressure conditions with pure oxygen. More recently, a fellow named Tom Schaefer has investigated total liquid ventilation, again replacing all the gas in the lungs, and has investigated this for inducing hypothermia. And he had a lot of problems with this. He had tremendous accumulation of carbon dioxide in the blood, And his cooling rates were nothing near what was theoretically predicted. And he didn't really understand why these two things were happening. And we didn't either until about 3.5 years of research later. And during the period of time that we were doing this research, another modality arrived on the scene, which was very instructive for us and helped us a lot, and that's called partial liquid ventilation. And what you do with that is you just put some liquid in the lungs, and then you just ventilate with gas, and you let the normal breathing in and out of gas do your gas exchange for you. And you can cool with that, and that was the first strategy that we used. If you want to load a large volume, about 60 cc's per kilogram, into the animal really, really quickly, you can cool about 2 degrees Celsius in a lean animal. Most of us, however, particularly in the United States, are not very lean. So in practice, we could only expect maybe 1 degree drop in temperature, and that's not enough. And furthermore, it's not going to be sustainable because you only get one shot at it. You put the liquid in and you can't get it out. Well, what kind of results do you get in dogs? Well, this is the result you get. If you load with 60 milliliters a kilogram of perfluorocarbon cooled to 1 degree Celsius, you get about a 1.5 to 2 degrees Celsius temperature drop. It happens really fast. It happens in about 180 seconds. That's quick. If you're going to try to rewarm using this technology, which is far less of a market, you got a lot of problems. You can't heat nearly as much. Your delta T between, in other words, the difference in temperature between the liquid you put in to warm with and the animal's body temperature can only be about 5 degrees Celsius, or you fizz. You get nitrogen bubbles forming because the nitrogen is really soluble in cold body fluid, and when you warm cold body fluid up, bubbles come out and you get the bends, just like deep sea divers, except from cold rewarming, from rewarming from the cold. So it's not a very useful modality in terms of rewarming from deep hypothermia, but it's still better than the conventional methods of external rewarming, and it's more applicable acutely than bypass. I need to give you a quick tutorial here on lung volumes. Functional residual capacity is about 30 cc's per kilo. Tidal volume is how much you breathe in and out. That's about 20 milliliters per kilogram under normal condition. And your total lung capacity is the amount of volume that you get out of your lungs when you squeeze all the air out that you, squeeze all the air into them that you maximally can when you take the deepest possible breath. So that's about 90 to 100 milliliters per kilogram. So what we're doing with mixed mode ventilation is we've taken the best of both total liquid ventilation and mixed mode ventilation, and we've added them together. Because we found with total liquid ventilation that our animals did not get very good heat exchange. They had terrible carbon dioxide removal, which would be lethal. And we're also, one of the reasons we can't get very good heat exchange is that we could only do 5 breaths a minute, because liquid is about 1,200 times more viscous than air, even fluorocarbons, which are very non-viscous. So the combination of mixed mode ventilation, which I'm about to define here, allows for addition and removal of the perfluorocarbon from the lungs cyclically. It optimizes heat transfer, and we didn't understand this until about 6 months ago, by inducing stirring of the liquid in the small airways. There's tremendous turbulent flow which breaks up the laminar flow that's normally present and allows for good gas exchange. And it allows us to ventilate independently of gas or liquid cycling. We don't have to be tied to the 5 breaths or 7 breaths a minute of liquid that we could normally move in and out. And it prevents hypercapnia. It prevents the accumulation of CO2 removal. It allows us to remove about 80 milliliters per minute at standard temperature and pressure of carbon dioxide, which is way over what you can get with total liquid ventilation. How do we do this? Well, we load to the tidal volume at about 50 cc's per kilogram per minute of perfluorocarbon. We then suction it out as fast as we can down to a pressure of about -5 centimeters of water. And we cool our fluorocarbon before we load it to about 2 degrees Celsius. And we ventilate in a special way with gas. And we vary the gas ventilation rate and volume at whatever we need to do in order to achieve control of our blood gases. And it's very simple. All you need to do is pass an endotracheal tube, blow the balloon up on it. You pass another catheter down the tube. And then you can add your fluorocarbon, and you can suction your fluorocarbon all from the same tube that you pass down the middle of the endotracheal tube. You just keep ventilating with gas with a regular gas ventilator. Very simple. Not so simple in terms of implementing all the valving, and this is the system as it exists. You've got a computer that controls it all. as of a few days ago. Prior to that, the computer was me, Steve Harris, and Mike Fletcher. You've got a cold reservoir of fluorocarbon. You pump it. You filter it twice, once with a pre-filter, again with a final filter to get rid of any bacteria or small particles. You run it through a heat exchanger, put it to an oxygenator, and then you deliver it to the patient. And you got a bunch of valves up here to control delivery and removal. When you want to remove it, you suction it back to the reservoir again and run the cycle over again. Now, this has been some of the neatest work I've ever done in my life in terms of just seeing the physiology come out of this. And we owe all of this to Sandra Russell. This is what happens in terms of temperature descent when you cool a dog using this technique. And this is a 5 degrees Celsius dog. He's part of our statistical run. We're 3 dogs into our statistical run, which will probably be between 6 and 9 animals total. And you can see that in the first 4 to 5 minutes, we have achieved a brain temperature, and the tympanic temperatures, incidentally, are normally markers for brain temperature, but we find in dogs they don't track very well. What is truly the probable brain temperature is the temperature between the central venous temperature, which is the blue line here, and the red temperature, which is the aortic temperature, which is the red line. And so what you can see is every time we load cold fluorocarbon into the lungs, the temperature goes way down. And then very, very quickly it recovers. This is arterial blood. So this is the blood coming out of the heart, right after it's been through the lungs, and the temperature goes way down with each load and then pops back up again. And in the venous blood, these tremendous excursions are barely perceptible, and they're more perceptible in this animal, which is atypical, in the rectal temperature probe, and the tympanic temperature probes, which are probably sitting in air potted with wax into the ear, swimmer's ear wax. They're very smooth and considerably warmer. And I'll explain more about what these mean versus what the real brain temperature is. So we're actually down 4 degrees, 3 degrees in the first 4 minutes with this technique. And all you need to be able to do is intubate. When we first started doing this, we had a lot of learning to do. One of the things that we discovered was that as we put more and more liquid into the lungs, and what you see in the green line here is just the temperature curve in the blood, in the aorta, in the arterial blood. So this is a marker for the liquid load going on. At its lowest level, the maximum amount of liquid is in the lung. And at its highest level, we've unloaded the liquid from the lung. And the other measurement here is the peak airway pressure. This is the actual waveform of each gas breath we give. And as you'll notice, as we get more and more liquid in, here, our ventilator pressure, our gas pressure goes way up. And this is very damaging, this top peak. And it doesn't buy us a lot either in terms of gas exchange. And you'll also notice that when we're suctioning liquid out and removing it, that our pressures go negative profoundly, very, very quickly, too fast for us to control easily by hand. We've since got that under machine control. And this profound suction, this negative pressure, is really injurious to the lung. So we've solved all those problems now, and this is what the typical curve or typical data set looks like from an animal. We don't have these peak pressures. The previous scale, to go back to it, is 30 millimeters of mercury peak. And this scale is, our peak is 30, and as you can see, our peak ventilator pressures are only in the 20 millimeters of mercury range, and we intend to chop that off even more on these last breaths before we stop loading with fluorocarbon. And you'll notice that we have eliminated any deep negative pressures. We're less than 2.5 to 5 centimeters of water negative pressure, which is fine. That causes no injury at all. I'm sorry, I'm going in the wrong direction. This is just an overview of what the gas breaths look like. You can see before the experiment starts, and you can see how things change when we load and unload liquid into the lungs. Well, how well do we do with this technique? Well, I'm not going to go over all the math with you. This is largely Steve Harris's work. People have long said I can't do science because I can't do math, and that's not strictly true, because I'm smart enough to know I can't do the math. So I get people who can, and they're just as important as anybody else, in fact, arguably more so, because this work is the foundation on which this whole thing rests. And the bottom line is, if you figure out the theoretical maximum that you could get, you should be able to do about 0.4 degrees Celsius a minute, theoretically with the fluorocarbon heat capacity that it has and the dog heat capacity and the load and unload rates and times and volumes that we're using, and our actual cooling rate that we're achieving consistently is 0.36 degrees Celsius a minute, half of the maximal rate you can get with cardiopulmonary bypass. And we can do this over quite a temperature drop. We can go from 37 all the way down to 27 degrees Celsius with this technique and have the animal survive running around just like normal, none the worse for wear the next day. There are some physiological changes that are associated with this, and I'll go into those shortly. This just shows another animal with a 10 degrees Celsius drop, and that was achieved in about 4,800 seconds. To go back to the previous slide, one thing I want to point out here is that the reason that we've done some of these 10 degrees Celsius drops is we've been interested in establishing the efficiency of the procedure, and I'm going to go back to even a previous slide. We wanted to know how fast the fluorocarbon is equilibrating with the body temperature inside the animal. So in this experiment, we start out with our loads fairly far apart, and what we see is that no matter how far apart they are or how close together they are, the temperature of the stuff coming out is always about the same delta T from the aortic blood temperature. So we're achieving essentially complete heat exchange with the exception of the dead space of cold liquid that remains in the animal's large airways. And even if we increase the speed at which we infuse and remove the liquid, we're still running the same delta T. So we haven't hit the wall yet. We have not yet upped our infusion rates high enough to find the point where we start having these temperatures come out far colder than they are shown here, and the spread between the green line, the top of the green line, and the bottom of the red line increase. So there's room for improvement. Well, again, to go back to the previous slide, and I apologize for my disorganization here on that, why do we see these excursions in the aortic blood temperature up and down with each load, and we see far less excursion in the venous temperature, and more to the point, why do we see this aortic temperature and venous temperature rebounding and coming up from their nadir here at 4,800 seconds back up about half a degree to a degree? The reason for that is that we are not uniform things. We are divided into three compartments in our model. The first compartment consists of the fluorocarbon in the small airways, the blood volume, the lung parenchyma, and parenchyma is misspelled here, and the endovasculature, which is the cells that line your blood vessels. And that is about 17% of your total heat capacity. And that represents the red excursions that you see. That comes to equilibrium very, very quick, within a matter of seconds, in fact. The central compartment is the well-perfused central tissues of your body. That's compartment number 2. That's your brain, your heart, your kidneys, your gut, which is not as well perfused as your brain and kidneys, but is still fairly well perfused, and your liver. And that accounts for about 72% of your total heat capacity, and that comes to equilibrium pretty quick. In fact, the blue line, if we go back to the previous slide, that represents equilibrium in that compartment. And the final compartment is the poorly perfused peripheral tissues, your gut contents, what's in your stomach and intestines that you've eaten, muscle, large airways, and your peripheral tissues, your fat, bone. Those do not come to equilibrium very fast because they don't get a lot of blood flow. So it takes time for thermal equilibrium to occur in these, and that's why you get the rebound. So it's important to go about a degree to half a degree under where you expect to be. Well, how are we doing with this in terms of the physiology? The answer is pretty well. This is end-tidal CO2 and minute CO2. This is how much carbon dioxide you make on the blue line in a minute during FC cycling. And what you see is that we're doing quite well. Compared to total liquid ventilation, we're doing excellently. And if we want to get right into the bottom of it, if we look at our carbon dioxides, what we see is that we're overventilating our dogs a little bit before the experiment, and our CO2s go back to normal, which is about 45, and throughout the whole length of the experiment they remain normal, and then we are overventilating them again at the end of the experiment and they come back down. And we see that we're delivering plenty of oxygen. These 3 dogs are from the statistical run. The previous 3 dogs that I showed you with CO2 are also from the statistical run. We've done about 15 animals using this general technique, and they all fall about the same in terms of the results. And we see a modest decline in pH, and that's a result of the fact that we're overventilating them previously. And when we restore normal capnia, we get, and there's normal carbon dioxide levels, we get a dip in pH, and it's transient and goes away as soon as we stop the procedure. If we were normal capnic throughout, we probably would not see this dip. In fact, we do not see it in some of the other animals that were done where we had tighter control of our gases. Well, another measure of how well you're doing is how much oxygen is left in your blood after it's passed through your tissues. And that blue line up here is called your central venous oxygen saturation. And as long as that's above basically 80, you're in great shape. And you can see that throughout this whole run, the SVO2 was fine. This is the worst single run that we have done with SVO2s. Most of our SVO2s are a straight line across with almost no deviation. So we're doing great in that sphere. One of the things that we noticed as a result of Sandra's work was that we saw that when we loaded fluorocarbon into the lung, we had a drop in heart rate that was transient. And my first thought on looking at this data was that the heart rate was as a result, the drop was a result of very cold blood coming out of the lungs and cooling the heart and slowing its metabolic rate or causing its blood vessels to constrict. And I asked Steve Harris to do an Arrhenius calculation to see if the amount of cooling that we could expect with each one of these loads of fluorocarbon of the heart acutely was causing the slowdown. And he found that it didn't match up very well. And Steve then looked at the data very carefully, and he found actually that what did match up really well, I'm going to skip that slide, was central venous pressure and heart rate. And you can't see that from this sort of data here, and you can't see it from data you would collect by hand because you're not fast enough and you're not good enough. This is computer-collected data, and there's heart rate. And if we were to remember the previous slide where the heart rate dips up and down, you can't even see it here. And yet, if we blow that data up and look at it very closely, we see that heart rate corresponds with central venous pressure. Every time the CVP goes up, the heart rate goes down. And that corresponds with maximum liquid loading of the lung. And Steve also solved this problem in explaining what was going on, and that is that there's a thing that you can do to yourself, that you do to yourself from time to time, called the Valsalva maneuver. And any medical examiner's office has a filing cabinet stuffed with photographs of people who have died while on the toilet. And the reason for that is when you hold your breath and strain powerfully, you decrease your venous return to your heart. And if you have coronary disease, that can be enough to tip you over the end and cause you to have a cardiac arrest. And that's called the Valsalva maneuver. And when we load heavy fluorocarbon into the lung, because the fluorocarbon has twice the density of water, we increase the central venous pressure tremendously transiently, and that causes our heart rate to drop. And if we put all this data together, again from computer acquisition, this is what you see. This is mean arterial pressure. This is central venous pressure, and this is essentially our liquid loads as monitored by temperature, and there is beautiful correlation between these. Mystery solved. Is this drop in heart rate and mean arterial pressure enough to give us concern? The answer is a qualified no. It probably is not enough to give us normal concern, and it is not the sort of thing that we expect serious problems with in clinical application. How are we doing? Well, we're 3 into the statistical run, and these lungs actually look pretty good. They're fixed. They've been removed from the animal, obviously, and you're looking at them from the back. The structure which runs down the middle there is the trachea, and there's the esophagus right behind it. We do see some isolated areas of injury, almost all in the bottom, most dependent part of the lungs. There's the trachea right there, so that this is the bottom side of the lungs. The heart is on the other side. And it's the areas that are the most dependent, in other words, the lowest down in the animal that we see this injury in. And it decreases our PaO2, or in other words, the amount of oxygen delivered to the animal, by about 10 to 15% for the first 12 to 24 hours after the procedure. We believe that this is due to acute chilling injury from the very cold liquid hitting the lungs, and we have a strategy to avoid that. although the damage is not overwhelming and the animals show no clinical signs whatsoever of injury. They are running around giving us great grief. In fact, we have to anesthetize them to do blood gases on them with an arterial stick. If we look at their lungs under an electron microscope, we see absolutely nothing wrong with the architecture. And the same is true of light microscopy. This is completely normal alveolar structure. We see nothing wrong. On light microscopy, the only thing that we've seen on some of the animals is modest leukocyte upregulation and more leukocytes being present in the large airways, surprisingly, not the small airways. But we don't see any red blood cells in the airways. We don't see any edema. We don't see any of the signs that would give us great concern. So, in conclusion, what we've got is a technique that, if you can automate it, any paramedic can do, and that allows for 0.5 degree Celsius a minute non-invasive core cooling. It permits maintenance of normal carbon dioxide levels and the restoration of normal carbon dioxide levels following ischemia. It allows for 4 times the basal metabolic requirement for oxygen, and it gives you near the theoretical maximum needed by a hypermetabolic patient of about 6,000 calories a day. And incidentally, to cool a human being, a 70-kilo human being, by about, say, 4 or 5 degrees Celsius is the equivalent of removing the explosive energy of about 1/2 pound of high explosives, or the equivalent in calories of 1 jelly donut. And that's a lot of calories in that jelly donut. It's a lot of heat to move in that short a period of time. In addition to superior CO2 removal, it allows us to far surpass what can be done with total liquid ventilation, which Tom Schaefer pioneered and patented for hypothermia. The best he can do is about 58% of the resting metabolic rate. This is not going to work for a sick person. We can get easily 10 times that. What we need to do now is create this. And what this is vaporware, actually, at this point. But the engineers who have built us our prototype system for delivering liquid ventilation in a more reproducible and certainly a less stressful and personnel-consuming way envision that this is what our final device will look like. And they actually have the schematics laid out for the design of this thing, and it would be fieldable. And I think that that is a potential profit center, and I think it can save a lot of lives, and I think we may be in clinical trials as soon as 2 to 3 years from now. Thank you very much.